Hello, everybody. Oh, that really picks up pretty good. Thanks a lot for coming to another exciting CDCR seminar where we have a special guest actually coming to us from industry, which is a bit less usual for these talks, but we thought it'd be great to give you guys that perspective. Dr. Cristian Draghici, coming from Novartis, where he's a principal scientist. Cristian actually was born in Romania, moved to the United States at the age of 18. In the Moldovan hills, which are in the northeastern part of Romania, I think a very safe distance from Transylvania. Healthy distance from Transylvania and anything that might travel as green mist. He graduated from Western Connecticut State University with a Bachelor of Arts in Chemistry and Biochemistry. He finished his doctoral studies at the University of Vermont in the Brewer Lab, developing synthetic methodology for fragmentation of cyclic carbon-carbon bonds. He began his postdoctoral studies in the Njardenson Lab at Cornell University and the University of Arizona, working on the total synthesis of vinagre. After his Sonoran desert adventure, he made his move back east, where he pursued postdoctoral studies at the Yale lab, at Yale in the Spiegel lab, where we did a couple tours together, and focused his efforts on the total synthesis of glucosapane, an advanced glycation end product. So that was a really heroic synthetic effort to build tools to study these end products, and actually earned first author science paper, which is pretty impressive. Now he's a medicinal chemist, as I mentioned. He lives out in Boston. He started off at the Center for Development of Therapeutics, CDOT, at the Broad Institute of Harvard and MIT, and then moved to Novartis at present day. So without further ado, it's my pleasure to introduce Dr. Draghici.
Thank you, Anthony. It's a pleasure being here. I haven't done one of these in a very long time, so bear with me. Had a lot of great interactions. Really enjoyed being back in an academic environment, talking to the PIs, talking to the students. I mean, talking to the students portion was really my highlight. And I think it comes from the fact that we spent some time talking a little bit about what should I cover here, what would be useful to learn. So certainly the drug discovery part I can talk about, but I thought that sharing a little bit of the experience that I had, sort of transitioning from the academic training into the industry could be a useful forum to discussion, and our conversation at lunch definitely reinforced that. So that will be part of the talk. And then the second is just basically what my first project at the Broad Institute in the Center for Development of Therapeutics. So this is taking a fragment-based approach to target small GTPases. So for that portion, I'll just preface it ahead of time, because a lot of the project is not disclosed. It's not in the public domain. I can't really share a lot of details about chemical matter and particular targets and whatnot. So sort of stripped it down to all that information. But it's grossly based on the job talk that I gave when I moved from the Broad Institute to Novartis, right? So I'm going to talk very little about sort of my experience in Novartis and things there, but most for the earlier part. So without further ado, so I just figured I'd start with a little bit of my synthetic calling card, if you will. So this is my training. It started at University of Vermont. So really here focused on natural methodology and natural products. So it's particular a fragmentation reaction that revealed these alkyne inoates, and then just sort of the use of these intermediates for more complex structure synthesis in some natural products. So this was like really formative years in trying to understand sort of the principles of what it takes to like develop methodology and work in a young professor lab. And then from there, I decided to do a postdoc and moved into Jorn Njardensson's lab. And here I worked on the total synthesis of vinagre. This is sort of a complex polycyclic natural product, and where really my contributions were sort of developing a couple approaches to form the carbocyclic core of this molecule, and that really served as the springboard for really finishing the total synthesis by other members of the lab. Another thing that I think it's worth highlighting here is in the Jorn's lab is probably most known for the drug poster. I saw it in Jake's lab as well. So the drug poster 200 most, blah, blah, blah. And really he has a lot of outreach that's involved in. So please look up his website. And besides the research, one of the things that he put out, and I worked with him when I was there, is the Chemistry by Design. So this is just a sort of an app that you can download on your phone, and you just like card form, you can just look at synthetic steps one by one and just kind of look at natural product syntheses. You can also look at sort of drugs and so on. So this was a nice, really contribution there, and I enjoy working on that. Then from there, decided to do a second postdoc and went to Yale. And this is where I, you know, as Anthony mentioned, that's where we met, worked together for about 3 years. And here I focused on the total synthesis of glucosamine. It's a really interesting natural product. It's formed in your body. It's an advanced glycation end product. It's a lot of very interesting proposals in terms of what the biological implications of these molecules would be, but they really hinged on being able to make these molecules. And they possess this kind of unique diaminoimidazole core that really was my job to try to figure out how to make it. And there we were able to develop, at least I think it's a neat little synthesis using a 3,3 sigma tropic rearrangement to assemble the core pretty quickly. And then somebody else's job was to basically take this on and develop some chemical tools to try to see where these are be prevalent and relevant. Last I heard, so some of this work is licensed, was patented and then licensed out to a small company that's interested in following up on some of these, but that's basically how much I know that's in the public domain at this point. So very, very interesting, but this is really where maybe a lot of folks here find themselves at this point, are close to, where, you know, I spent a couple times, couple postdocs, and my whole entire career up to that point is really spent working in PI's labs that did not have tenure. So it was, you know, the beginning stages. It was, I mean, it was tremendous learning because you're helping set up things and you really build confidence and be able to like set up projects, think about what it takes, high stakes, you know, it's a lot of like new learnings that ends up published. So that's really cool. But at the same time, you do kind of get a glimpse of what it takes to set up a lab and be a PI. And for me, especially with 2 postdocs, was a lot of consideration whether or not I would go in that direction or not. And it just really took some time for me to figure out that academic path would be something that I'd be interested in, but is there something else out there, right? So I just put this crude 2 images, you know, I think, unfortunately, ends up that a lot of times you're just left with this like binary decision in terms of like going academics or industry. And I don't think it's necessarily right? And there's a lot of misconception about industry, and I didn't know anything about it. I mean, really, like if I put this picture there, it's really all I could think of at the time, right? It's just like shiny object, big, lots of interesting things might be happening, but I didn't know anything about it. And then something actually that came up today, and this I had on there before, right? It's this idea that like somehow the industry is like pulling you to the dark side, something that's like out there. And I think it's the unknown of it. I mean, this is a gross oversimplification, right? So this is for dramatic effect, but this came up today in conversation. I think there's reality to this, and I don't think it's a good reality to accept. I really think industry is a complex environment, right? And we define as 2 binary things, but you know, academics and industry. But there's a lot of things that go on in industry. There's small companies, biotech. There's venture capital. There's all sorts of things that you can be involved in, which your job or your training as a PhD can have application in. And not having that exposure, I think, is a detriment in some aspects. And I think there's still lots left to do to kind of demystify this process, both on the industry side, but also I think on the academia side, to just kind of bridge the 2 and just kind of provide more information for really the students and future workforce to have more information about the choices they're about to make, right? So, you know, for me, I feel like if I share my experience, that could serve as a talking point, and feel free to stop and ask questions or discussion points. I mean, there's some room to talk about that. But for me, really was what I couldn't appreciate was that, and it took me going into industry to really appreciate this, is that there's a lot more going on in industry than we know about. And the decision to go to industry is not just I need a job and go into industry, but there's a lot more. There's a deeper problem and questions you might ask yourself. Where do you want to live? What kind of lifestyle you want to have? Who do you want to be surrounded by, and so on. So there's, and I think you need to think about that when you make that choice and moving into either industry or academia. And I'll leave it there in terms of where I'm at with it, but there's definitely a lot of room, I think, for improvement there. So conversations, talking to people that work in both places, trying it out is a way to go about it. But spend the time. And if you can spend the time before you have to graduate, spend like 1 or 2 years preparing yourself for that process, I think you'll be in a position where you can make a better choice. So yeah, just kind of to give a little more granularity to the binary problem decision between academia and industry. So I pulled some statistics. These are this is US related, right? In 2019. So this is NSF research. And really what it's showing you is just kind of the job distribution in between academia, government, and industry, right? So this is one of the problems, is we define it that way. That's just a definition. But there's actually quite a bit that actually I thought industry would be a higher, but there's quite a bit that is actually qualified as academia. And it's quite interesting to look also how the differences are based on the type of training that you have, looking between chemistry, genetics, or just pure biochemistry or whatnot, right? So maybe spend some time looking at where Canada or US and trying to, that's one place to start to get to prepare yourself, kind of what area would you enter in and where do you fit? I mean, government could be good, or just staying in academia. And academia, I think at this point is not, at least in this graphic, does not just encompass like a PI position, because those are probably only about 5-10%. There's just a lot of other things that you can do within an academic environment. And this is really kind of where the broad job came in for me. This graphic here, I think it's interesting to look at because, so it's binary or ternary, whatever, industry, academia, but really when you look within those areas, you have a multitude of different jobs you could be doing, right? So it's not just R&D. That's how I was thinking about it, right? You just go and you have a lab job and you make them all because I'm a chemist. That's really what it was, but it's more than that. And once you enter the system, you actually get to see all of this, right? So you can work in, if it's academic, it could be research, it could be teaching, technology, transfers, and all this stuff like that. And also all of this stuff happens in the industry as well. So there's a multitude of types of jobs that you could be doing, again, with your PhD level qualification. So I think, I've only pulled these down like recently because I was curious about this as I was coming to give this talk, but I never looked at this when I was in PhD, when I didn't even think about the diversity that's potentially out there. So it's something to think about. All right. So, for me, again, retrospectively, I look at this and then I got lucky and I was able to transition to the Broad Institute. Now, the Broad Institute is for my first job. Broad is an academic institution, is a non-for-profit organization, and it's essentially a collaboration between MIT and Harvard, where they get to collaborate and work on joint projects. I mean, I think it's a lot of similarities to what you guys are trying to set up here, right? And McMaster has potentially done already with other endeavors. So to me, it felt like a good compromise where I could still have a little bit of the academic and be connected to academic labs, but then also make a transition into a staff scientist position, which is what I was hired at in the Center for Development of Therapeutics. Now, the way the Broad was set up is you have the PI labs, which are core institute members. There's also affiliated members. And then within the PI labs, there's a lot of genetics, a lot of biology, and it would be targets that would be interesting in working in. CDOT would offer the sort of know-how and drug discovery technology to try to enable these programs to move in towards full-fledged drug discovery programs that would end up in collaborations with either pharma, or venture capital, and so on, right? And to me, it really felt like a really great compromise to be able to start and learn about drug discovery in this format. It's not a lot of places like this, right? So it's not like there's a ton of options. And I got lucky with this in my particular situation. But that doesn't mean that there's not other options that one could take as they make the transition into industry, if you will. And the projects that I'll be talking about today are going to be essentially, these were projects that were collaborations with Calico and then Deerfield. So quite different. Calico is an offshoot of Google. So kind of like pharma, small pharma. Deerfield is just a pure venture capital. This kind of opened up a lot of new ways to think about what it is to take to fund research within these kind of institutes. So this you'll find quite a bit, again, something I didn't know anything about at the time. I just learned it being part of it. But for any biotech or pharma or anything, there's a structure in place, right? And in this case, you have projects that are being supported by various disciplines, if you will. So you have Medcam, pharmacology, protein sciences, structural biology, biophysics and chemistry. Big part for us was also the PI labs that would come in with their own technology, and then alliance management, which played a huge role in setting up the collaborations with the different funding agencies. And also a lot of the work that was done with CRO. So my project was, I was the main chemist, and then we had CRO resources where we'd execute on the chemistry. So we do the design and some of the chemistry ourselves, but the bulk of it was done with the CRO. And eventually, even within all of these, you would, if you get to a point where you've optimized an assay or something to a level that's transferable, you would bring it to a CRO and be able to execute it on scale that way and free up internal resources. Now, with this also is to keep in mind that, so this structure is also what's used to set up teams, project teams, right? So in general, what you'll have in a drug discovery program, you'll have project team lead, and they could be from biology and chemistry. It's generally set up based on the needs of the project at the phase that they're in. And these projects then will bring in from the different areas depending on needs. They can be larger, smaller. If you're early in a drug discovery program and you need to set up a protein and do the expressions and everything, you would have more protein sciences, structural biology. But in general, you'll find in a team structure, you'll have a biology and chemistry lead running the projects. And that was true at the Broad, definitely true at Novartis as well. And these teams would then be, you're charged with basically taking projects, in this case would be starting from target ID and validation, which would be executed primarily coming from the PI labs or some other target that's well disclosed and you're interested in working on. But primarily were novel targets that were coming from the Broad PI labs. And it would try to do this is standard, you'll find it everywhere, the different discovery, stage 1 and stage 2. Obviously, given on the amount of resources and people and knowledge that's available and experience, a lot of the work that was done at the Broad while I was there and probably continues now is up to lead ID, right? So here's where you have your target, you have it validated, you know it's relevant for a disease, you initiate your screening campaigns. I know you guys do screening here as well, and then you try to optimize those to lead-like molecules. Lead up, it's something that requires quite a bit of resource, chemistry, and also PK sciences, in vivo pharmacology and whatnot. And that's a lot harder to execute at a place like the Broad. They were interested in doing it, but this is really where the alliance management collaborations would come into place, right? So if you have something that's getting close to that area, then you start to bring in collaborations and partners to try to execute this and bring it into stage 2, which is essentially the preclinical development and certainly all the other portions that you have to do, clinical trials and everything else. So this is really something, I mean, when I look at this, really, this was part of the decision for me to move from the Broad to Novartis, right? So I could see a lot of this at the Broad, but for me as development as a scientist and medicinal chemist, drug hunter, you'll hear that term too. I'm not sure I quite conform with that yet. But it's really hard to see stage 2 type in an academic environment. If you want to learn it, if you want to see it, you want to see it applied across not just small molecules, but all sorts of different technologies, you got to go to a place, in my opinion, that does it. So that was part of the motivation to go to a place like Novartis. So this is pretty standard. I'm going to transition now more into you know, the sciencey portion of it, and just kind of what the first project was at the Broad. So if there are any particular questions about this or discussion, you know, maybe this would be a good time to do it. Have a little break.
Out of curiosity, since you've been across the spectrum, how did you feel that your work-life balance changed from a postdoc and to Novartis?
I mean, I think you know the answer to that question. It's drastic change. Yeah, it is. I would say, you know, the workload and stress level is, I would say, similar. I mean, full disclosure, I'm also in a very different part of my life. I have kids. That has a whole other level of stress that's not work related. So I think in terms of like the stress levels and, you know, the drive to accomplish things, this is the same. That doesn't change, you know. My hours have changed quite a bit, but I think you also learn to get a lot more efficient. I don't know that I was as efficient as I am now in some aspects when I was in grad school. I just felt like I had more time in grad school to do things, to stay 12 hours in the lab and whatnot, right? I mean, I'll be fully honest, I know people that were in the lab 12 hours present, but not necessarily doing much, right? So I think in the industry environment, everybody's very motivated to go in. You're there in your hours. I mean, we don't punch cards, you know, nobody waiting for me to walk in at 9 o'clock and leave at 5 o'clock. We have freedom to, you know, we're also now post-COVID, so work in an environment where there's hybrid work environment. If you need to do stuff at home, you do stuff at home, you know, but it's about executing on your tasks. You have a project, you have deliverables, you agree as a team, this is where the team sport comes in and what you need to do, and you got a job to do, and sometimes you need to stay a little longer because you need to get it done. You just do it. But there's nobody whipping up, cracking a whip, if you will, trying to get you there. So it's, yeah, it changes. What's that? No. Any other? All right. Take your time. Yeah.
I have a question that might be of interest to some of the students.
Yeah.
To what extent do you think they have to train specifically in an area they want to go into? If they want to get into something like biophysics and biochemistry, you know, could they have more of a Medcam background and still be competitive for a job like that? Like, what do you think? What should kind of guide them at the PhD stage? They know they're in the industry.
I mean, it's hard to argue that you're trained in Medcam and you're going to take a position in biophysics if you have no experience in that. I think if you could show that you've done it, right? And I think the collaborative places now in academia where you can actually do from synthesis to testing molecule, and you actually get exposed to biophysical methods and biochemical methods and developing assays and whatnot. So I mean, chemical biology is certainly a good training ground from that, because it kind of forces you to do both. But yeah, I mean, I think in general, you want to apply and you will be considered for the skill set that you have that's appropriate for their, you know, company's needs, if you will, you know, so.
So some overlap, but you don't have to be a specialist.
Yeah, no, I mean, there's room, and, you know, it also depends on where you apply, right? So that's like big pharma will have more specific roles they try to fill, but if you go to a small biotech, fewer people, you have to wear a lot of different hats, and a multitude of skills could be more appreciated because they would benefit from that, right? But overall, I'd say, you know, skill set is important. You should definitely advocate for your strong skills. And I mentioned this in lunch today, but I think it's really important. Besides your technical ability and knowledge of science, another thing to keep in mind is that when you go into industry or any job interview, what people look for is also like, can they work with you? Like social skills, the ability to interact, the ability to problem solve, right? I mean, I've said this a bunch of times. The likelihood that you'll work on something you worked in your PhD or postdoc is, I'd say, zero, or very, very small. So you're going to work on something that's a different target, a different disease area, something that's completely different. So your ability to problem solve becomes more important, right? So can you be given a task and know how to execute in answering that question? I think that's really, really important, right? And you have to try to make that clear during your interview process. And then people try to fish that out through questions, and things like that. So it's something to keep in mind. It's hard to train for these things. I only saw these once I was in and I was interviewing people. So practice is always good. Mock interviews, or just apply for jobs and go through the process and just try it out, see what works, see what doesn't work. It's a big part. And that first job is the hardest to get, right? Once you're in the system and you've kind of created that space and you understand that a bit more, it gets better. And that's probably true in academia environment as well. It's just, there's a process. You kind of have to learn how to lead through it. Yeah.
I hope you don't mind. I'll ask a question for the students as well. What's the starting salary of a PhD chemist or scientist?
Canada? I don't know. In the US? It varies. So, I mean, it's over 100,000. I mean, I'll just frank, like at the Broad is a non-for-profit, so it's lower to begin with. We don't have bonuses, so it's structured a little bit different. I started at like 90, which, it's kind of on the lower end. But if you go in small biotech, whereas, there's basically higher likelihood of failure, right? But they still want to recruit top talent, they'll pay more. And then you can get like incentive bonuses at the end of the year, and you can get also, I mean, that statistic is out there. I could have pulled it out. I didn't think about it, but but no, it's I think the average 100,000 is pretty reasonable. So that's one is reasonable.
There's just some context. One interesting difference between undergrad and graduate education. Undergrad education, at least in Canada, usually often has like co-op options and certain codes out there, industry, government, whatever. At a graduate level, they're starting to have that, but it's not nearly to the same extent undergrad. So knowing that context, I was wondering, I think a bunch of students in this room might even have like, from several years ago, let's say, spent 4, 8 months, 12 months in this setting. By the time they graduate their PhD, the experience is probably 5 to 10 years old. So I'm curious, even if it's old experience, I mean, would that still help you stand out significantly for someone who has academic old experience?
Potentially. I mean, it really depends on what the people that are hiring are looking for. If it's something that they're interested in a skill set they're trying to bring in, that experience could set you apart. Any experience could be useful. But generally, I mean, I did 2 postdocs and I entered at level 0. So none of that academic, training counted as experience when entering the pharma world, right? And I think part of it is also, in truth, is I didn't know anything about drug discovery, right? So I was a level 0 in that sense, right? It's just what it is. So it takes, I think, good 3 years to just get your feet wet. It's just like learning a new language, there's new acronyms, new everything, and learning how to operate in a team, making decisions. And it's just a huge amount of learning that you have to do. So if you have that from other environments, so like some labs, like, Jake's lab does a little bit more of like the, Medcam type collaboration, sending compounds and like that. That's the system that works. That's good experience to have, where you can advocate that as like, you already know kind of what it is to operate. But I would assume that when you enter, you're going to be treated as freshmen. That's pretty common. Cool. Good question. Should we move on? All right. So this is like, I think it just serves as a good primer to kind of figure it out, where the project would fit in that I'm going to talk about. So at the Broad, there's a lot of, I mean, if you're familiar, there's like the diversity-oriented, Stuart Schreiber's there, so there's a lot of chemical biology, diversity-oriented syntheses, so some unique libraries, a few 100,000 compounds. David Liu is there now as well, so there was some work done to develop some DNA-encoded libraries. So there's a variety of different libraries, but one of them that we thought at the time within CDOT could be applied from a structure-based design perspective was fragment-based drug discovery as well. So that's what I'm going to focus on. So I figured I'd take a few slides here to just kind of, for those that don't know anything about it, maybe put a little primer on this. I think it's a really cool technique that you can apply in small scale in academic environments. And there's a lot of academia that's worked with these. And I think most importantly is that it's well established now to deliver compounds in the clinic, right? So I think it's been shown for very challenging targets, you can take a fragment and develop into a useful drug. And these are just some examples that I pulled in as of 2022, so that's last year. There's quite a few. And the technique is probably 25 years old or something like that, right? So it's still relatively new. So it made a sizable impact, as I'm saying there, in challenging targets. They were deemed undruggable, so like PPIs and things like that, not just like working with kinases, if you will, but even there it's made a nice splash as well. It's one of those that it's a technique that benefited from a growth in biophysical methods, right? So understanding how to use NMR, advent of SPR, mass spectrometry, and then also these technologies being able to be deployed on scale, right? So this kind of technique benefited a lot from that because you're dealing with small molecules that are weak binders. It's just the principal point of it. So you have to have ways to be able to characterize that. But because of that, and they kind of grew together. So the biophysics benefited from it because it can push the boundaries, and at the same time, it enabled fragment-based drug discovery to be able to do. So now people do some really cool stuff now. Where this was done for a while, where they screen directly in crystallography, and you can even do cryo-EM to see small fragments bound, which is super cool. So lots and lots of developments that I think it's a nice example in science where things grow together and they benefit from one another, particularly in this case, biophysics, chemistry, and biology. The other thing that I want to, so practical fragments, if you're also learning, let's say you want to go and or even consider about injury, there's tons of blogs, right? So I'm mentioning practical fragments. This one is just posts about fragments. It's good to read about this stuff. The other one that I found really useful, and I forgot to do it in here, is In the Pipeline. I don't know if you guys know this one. It's really famous. It's in the science, and he's at Novartis as well. He's a really interesting character, but he writes about a lot of different topics in pharma. So reading those, it'll help you prime and understand a little bit more about what goes on there. Another one that is used, Drug Hunter, drughunter.com. Well-curated website. They've gone to the point where they do some really cool analyses on just clinical compounds that are out. They have subscriptions for academic environments. And I'm not working for them to plug them or anything, but it's a really useful resource, right? And this, I took this image from them. And another point here, fragment-based drug discovery is like well-established. I pulled a lot of this information from just Google Internet, right? So if you're interested in something, just Google it. You'll find a lot of really useful information and references, and in this case is even like well-curated presentations about what it is to do fragment-based drug discovery. So why fragments? This really captures it, right? So the idea is that if you say you have 30 heavy atoms, the chemical space is just ginormous. This is an estimation 10 to the 63 of possible permutations that you can make, right? And your largest libraries run in the millions. You just can't cover that space. It doesn't matter how much, you just can't make that amount of chemical diversity. So you're really only sampling a small amount of chemical space when you do these high throughput screens. Now, that's not to say that they're not useful. It's just you can have other options. And in this case is really where the fragment-based drug discovery came from. Is that if you reduce the amount of heavy atoms you have in a molecule, now you've decreased the amount of chemical space, let's say 10 to the 9. And you're overall, with a relatively small few thousand compounds, you actually cover quite a bit of chemical space to give you some diversity. At the cost of affinity at it, right? So you're starting much weaker starting points, and that's just something you have to understand. And this is really all the biophysical methods that I mentioned come in handy. So the other aspect of it is you'll do high throughput screening. These are run regularly, right? Novartis has millions of compounds in their libraries, and they run screens a lot and various targets. But you have to go through a process of making sure that you it's the right target, you have the right screen, you can do phenotypic screens, you can do all sorts of different variations of the high throughput screen. But in the end, one of the key features that comes in is that you might find a compound, but it's quite well elaborated, 400, 500 molecular weight, and that's not really fully well optimized for that binding pocket. And actually, just kind of what this picture is trying to represent here, you can spend quite a bit of time trying to do chemistry, quite difficult chemistry, to try to get that perfect molecule. It has a good starting point in terms of properties, which is a key point, but also just suited for fitting that binding pocket in affinity. So about 50% of, I think I was reading this, about 50% of high throughput screens just fail on principle, not giving you quality chemical matter that's pursuable, right? In contrast, and I'm going to make this point now, is they're not mutually exclusive. It's not like you're going to do a high throughput screen or you're only going to do a fragment screen. You can think about these in parallel, right? And it depends on what the target you have and how you want to approach a problem. But at fragment, you start with smaller amounts, and these small fragments can actually fit and be optimized for your pocket a lot better, even though they're weaker. And the weaker affinity is one point, but that can be optimized as long as your ligand is efficient. And this is really what it's trying here. And ligand efficiency, I'll talk a little bit about this. It's a concept that's used quite a lot in drug discovery, right? So that is how much potency and bang for your buck you're getting from the amount of atoms that you're using. If you're using a lot of atoms to try to get potency, and particularly lipophilic, that's usually not a good place to go into. Again, something I didn't know anything about until you start working with molecules. And the nice part about fragments is that you have a weaker starting point, but now you can use structure and your chemical intuition and your chemical skill set to start to derivatize these molecules in a more rational way, and keeping in mind your property space. So you're not giving a starting point that's already probably in bad space for whatever reason. And let's talk about those reasons a little bit. High throughput screens or libraries are usually assembled from, compound collections, either commercial or things that you've made in-house over time. So a lot of them are made for a specific target, right? So worked on kinases or something like that, right? So that's one. So they become kind of focused on particular targets. They're not necessarily suited for different types of classes. And that just kind of speaks a little bit to the chemical space. The second point of it is that in that process, in terms of chemistry diversity, chemists got really good at using a few transformations. Actually, there was a paper, I forgot to put that here. It's a good one. It's a good paper that's in journal Medcam. Top 10 reactions using, can you name number one? You got it, right? And then the second one is, I think, Suzuki reductive amination. And the third one, I think, is Suzuki couplings, right? So very useful. But then in the end, you only end up with a certain amount of weight to put these molecules together, right? So you end up in a lot of flat space with Suzuki couplings and lipophilic and whatnot. And that's represented in these what you capture in these large libraries. A lot of them is just, that's why they're not suited. There's going to be too lipophilic. And that's just one aspect of it. Lack of three-dimensionality, right? So trying to include more of that. You go into fragment space, and then that diversity is a lot larger, weaker starting points, but then you can build that slowly in a more rational way. So that's really kind of the gist of what I understand from that. There's a lot more you can talk about here. So fragment space, like they usually obey this rule of three. You might have heard of the rule of five. I'm not a big, I use the word guidelines. I think they're useful to use. I'm not a big rule guy. And I think drug discovery has definitely moved into a space that's just like completely obliterated rule of five rules. I think they're still good to keep in mind, but PROTACs and all sorts of new molecules, just completely beyond that. So these are things that you would start with in assembling a library, for example, and I'll show you an example of what the Broad put together. And then these, I think, are critical, right? And they speak to the biophysics portion of it. Things to watch out for. And these, a lot of them are learned just reading through this literature, just learned over time by people assembling libraries, using these, right? So things to watch out: solubility. Solubility is really important because they're weak fragments or binders. You're going to screen them at very high concentrations. And if these compounds are not soluble at high concentrations, you're just not going to get good hits. Reactive molecules is an obvious one. I mean, you can have covalent libraries, and those are used quite a bit. Fragment covalent libraries. But then there's also a lot of like masked reactive functionality that can give you problems just covalent or reacting with your protein. Aggregators is an obvious one. And then nowadays, libraries are already screened for potential aggregation or aggregators are flagged ahead of time. And, so these are always good to keep in mind when you start working with fragments. This is an example of what the Broad library, early Broad library looked like. So you can go through commercial availability. So it's like 250 plus compounds, and you can select based on diversity, right? There's still some spots that can be essentially filled in a little bit more. So this is just looking at chemical space, but you do get pretty good coverage. And then the same from a chemical cheminformatics point of view is just kind of what you're representing, the molecular weight, log P starting points, and so on. So these are not difficult to do, and you can just essentially assemble it internally. And then you have a library. So libraries that I've shown, these are physical libraries, right? So these are things that you would have and screen, but you can have virtual libraries assembled as well, and those can be screened computationally if you have a good structure as well. And then you get into the screening methods, and these are all the biophysical methods that I mentioned, right? So the main one that we used and drove a lot of the program that I'm talking about is NMR. And you can do protein detected and ligand detected. There's full-on experts that know how to do this stuff. It's really, really, really neat, cool what they can do with this stuff. Crystallography I talked about, SPR. So these are standard ways that you can screen. But the key point here is that it requires stable and solubilized protein, right? So you're going to screen at high concentrations. You're going to do an NMR, so you need stable protein, things that you can purify, things that you can express on N15 labeled, and so on. So that requires you that part that I showed you before of having in-house ability to, you can outsource some of this stuff, but having it in-house and doing protein sciences is really, really crucial. And then the other one that I think it's emerged more is that unlike the high throughput, which is a better starting point in terms of potency, if you will, here you're starting weak fragments. And you have to invest in structural biology. You want to know how ligands bind. And two, you want to have dedicated chemistry effort pretty early, right? Because you can buy stuff, but you're going to have to be pretty methodical about exploring your fragment in directions you can go to. And for that, you need investment in Medcam. Unlike other, maybe from a high throughput spring where you bring it heavy chemistry a little later, you need this early, thinking about it. And the last point here, so you've screened, confirmed your hits, and then once you start growing your fragments and doing the chemistry, this is something else you'll see a lot in drug discovery talked about. We can spend hours talking in industry about how to optimize this, is the DMTA cycle, right? So how quickly you can turn the wheel from making a compound, analyzing it, and making the next decision for your next round of iterations. And the more you can do this, the faster you can answer questions, right? So optimizing this is a lot of work that we do in the drug discovery teams to make sure that we got that nailed down. All right, so how are we doing on time? Really poor. Okay. I talked too much. So let's dive. Okay, so fragment-based drug discovery. Now let's move into some biology. So the first project that I worked on was a GTPase. And there's about 150 of them. There are a lot of them that are relevant in disease. Aberrant functions drives disease, predominantly single point mutations, really relevant in cancer. The ugly cousin of this is KRAS. Everybody knows it drives about 30% of cancers. They're described as functional switches, essentially are active at the plasma membrane. They engage effectors and then turn on a slew of downstream signaling that control key processes in cellular function. Structurally, they're pretty small proteins. They have a G domain, so it's highlighted here in yellow, and two switches. These are the functional ends. And they basically function based on GTP versus GDP loading. So this is the switch from GTP to GDP, GTP on, GDP off. Really important class of targets. They're high flexibility, lack of obvious binding pockets, and this has made them notoriously difficult to target. I mean, we've been trying for, especially in the RAS space for 40 years to drug these proteins because they're super relevant to cancer and disease. So in terms of the bro strategy, taking this, right? So it's just a quick primer here. The idea is to take this switch, right? So it's constitutively on, turns on signaling. So we want to turn it off. So the idea was to focus on the on state. So when it's mutated, the protein is locked in the on state. So it can't actually hydrolyze the GTP to GDP. So the idea was to try to target the GTP form, which is the active state, but at the same time, taking some clues from the literature that describe that in the GTP active state, there's two kind of conformations. There's an active and inactive conformation, and one that has active signaling and the other one that has impaired signaling. And that we were able to distinguish these two through biased conformations that are induced by particular stabilizing mutations, right? So if we can actually target the GTP in an inactive state, we'd be able to target a predominant form that's available in the cell and be able to turn off all the downstream signaling. Now, these molecules are really, again, very flexible, so it's very hard to target them. But we thought that utilizing some artificial mutants that would be able to stabilize it in this inactive form would give us the entry points that we'd need to do to start the drug discovery process, and then after that, go back and see if it actually equates in the oncogenic mutant. So that was always the idea. And as I talked about, we're going to go take a structure-based drug design approach. The collaboration setup was all in-house, and I already mentioned all the pieces that would go into setting up the project, and then challenges, lots of controls, lack of controls. So it gives you no way to start point to set up assays, and that was really bad. That was really one of the first accomplishments, is actually putting some controls in place internally. Lack of assays, which ones the relevant assays they would go after. And I mentioned the shallow surface, no obvious binding pockets, so that's inherently challenges. We're not structurally enabled, and as I mentioned, that's really critical for fragment-based design, so we needed to do that. And we have to think about selectivity across all GTPases and isoforms, and there's a lot of homology within certain of them, so that's something that we'd have to keep in mind moving forward. The idea was to deliver single-digit micromolar inhibitors and hopefully demonstrate some cellular activity as a starting point. That's really kind of what marks that hit to lead and going into lead up, potentially transitional lead optimization. So the screen, all the methods that we decided to do NMR and mass spec screens. We screened our internal library. Really what we found is a variety of hits, really only one that cross-referenced in two different methods. But essentially, there were two privileged clusters of binders that were confirmed by both crystallography and NMR, and those are essentially our starting points. And they were used to essentially also triage our orthogonal methods, all of them. And we tried like literally everything we can get our hands on to see whether or not they would replicate, because we didn't have control. So we had to actually constantly keep an eye on that. So this is where the Medcam portion starts. I mean, this stuff can get dry and a little boring to some people that don't do this kind of stuff, these numbers. So I'll walk you as slowly as I can through this. But the idea is you take your hits, they're millimolar binders. There's really weak binders. And then you can do some SAR by catalog, just probing around where you're missing. You can do some optimization in chemistry, and that's the chemistry that you need internally. And we got them down to micromolar, 300 micromolar. And we started getting structural enablement, right? So now we get are potent enough that you can do crystallography for these ligands, so that you can start enable some structure-based drug design. And for both hits, these privileged scaffolds, we're able to do this. And started assembling this assay toolkit. Like I said, we're trying everything we can. This was a functional assay that allowed us to kind of have a surrogate way to probe whether or not inhibiting or touching this protein has a functional effect. The other thing that you'll see a lot, and this is a parameter that's used a lot in fragment-based drug discovery, is ligand efficiency, or LE, and I'm showing these numbers here. Ligand efficiency and lipophilic ligand efficiency and Medcam, these are constantly used. The idea is to track by what modifications you're making. You want the smallest modifications to give you the biggest bang for your buck in terms of potency, right? And the less atoms, more potency. These equations basically derive to a larger number, right? So in this case, you get 0.24, and the idea is to at least maintain or improve this number as a ligand efficiency parameter as you're modifying your molecules, right? Yeah, let's move on. So what do we do next, right? So we have a couple of fragments, and from the structure, it really became evident, and I'll show you some structures in a little bit, that we can actually start modifying these molecules and taking one piece from one and then one from another, and trying that, we actually started improving and then getting into the sub 100 micromolar. And at this point is really where we started to see some reasonable potency in our oncogenic mutants. So I'm labeling this A and B. So the A is this sort of mutated form of the protein that was more stable, and the B is the oncogenic relevant mutant. So we wanted to track this and make sure that we're moving in the right direction. So we're starting to see some functional activity and start to build this paradigm of affinity function structure, I call it. So it's you're binding to the protein, you have a functional activity, surrogate as it may be, but then how are you seeing any structural changes in a protein that can make sense in terms of what you're imparting in terms of function, right? So can you correlate all these and start build some hypotheses in what direction you might want to move with your compound to be able to get the desired functional effect? So just some blob structure here and structure. I mean, this is where it really gets into structure-based drug design. This is really the part that I really like. So this is the apoprotein. And it's nothing obvious in terms of what you'd call a binding pocket in here. But these areas of the proteins are those switches that I told you are highly flexible. And then you get your first crystal structure, and this is in these double mutant or mutant structure. That's a little bit more stable, and you can see that this just changed dramatically, right? And I see that opening up, and you start to think about, well, it looks like I have some room to move in that direction. I mean, it's not, rocket science. It becomes pretty obvious at one point, and it's just what you put in there, right? So libraries are very common. The first library I ever ran was an AMID library, and this is what this was. I've learned some lessons from that. But the idea here is to go to, we had the right building blocks, amines and carboxylic acids. We can quickly couple them. They were really successful, as you'd expect. And we identified some now micromolar binders, right? So we get down to the single digit micromolar and a double mutant, double digit in this single mutant or the oncogenic mutant, and also functionally active. So this keeps tracking as we're moving forward. So we have some interactions, potential for new interactions that we've identified. We have some novel receptor conformations as we're now growing this molecule. And also you're starting to understand what are the important parts of the molecule. I have them labeled in colors here just to kind of represent these, graphics. And each one of them starts to have an important role, right? So it looks like here I have some room to grow. And do I tolerate SAR? Can I start modifications there and they're tolerated or not tolerated? Can I put lipophilicity, polarity? What are the key things that I can do? And that all requires a lot of chemistry. So eventually with this particular series or sub-series, we ended up at a point where we had single digit micromolar, but overall the highlight here is that the property space in these molecules was not really where we wanted to be. And we started identifying some features in this where we thought some of the properties can be improved, and it was particularly this turn. We had really high PSA, polar surface area, solubility varied quite a bit depending on what kind of substitutions we had. So we thought we had some opportunities to go back and retool the molecule and give us some better property space. And this is really, we transitioned to a completely different funding source. So we had an opportunity to kind of reframe the project, which was a huge advantage here. The other thing that you will see a lot in drug discovery, and I had to throw this here, is workflow, right? So you need a flow chart. You need to think about how you're going to run your compounds through your assays. both in a biophysical or in vitro sense, but also in vivo, to get yourself to really see if you get the desired cellular activity, and do you have the assay toolkit to put that in place, right? I'm not going to go through details of this, it can get a little boring, but this is where you start thinking about what are your critical assays that will teach you the right information to move down the flow chart to get you to your cellular assay. And now with this in place, we're looking to move it from a cellular activity to actually deliver in vivo usable compounds. We never really got there. I left before all that was done. But really what I can highlight in here is just how the learnings from one series and all that structural information now become useful for us to like retool the molecule. And this happens a lot, right? Like you'll have a series, you'll have a backup series, and the more resources you have, the more you can carry at the same time. You don't put all your eggs in one basket. There's a lot of unpredictability. So it's good to give yourself options, and we did this kind of sequential. But oftentimes what will happen, you might be doing this in parallel. And once one starts to move in the right direction with a little better properties, you start to put more resources into that. So this series B essentially can we take this portion yellow of the molecule and modify with something else? And we did that. We just built 40 analogs, based on chemistry that we knew we can execute. And we got something similar in terms of potency and reasonable ligand efficiency. So now we needed to explore what else we can do here. We got pretty quick to molecules that are a lot simpler, a lot smaller, better property space with, single-digit micromolar potency, and all that structural information was critical there. The other thing that we discovered here is a whole new things that I had to learn about is atropisomers. So these are different type of chiralities, axial chirality. These actually start to become quite relevant when you start looking at protein-protein interactions, and there's, drug molecules that are atropisomers, and it's something to pay attention to. Like even amides can have atropisomers, and it's becoming more obvious. You can spend an hour talking about this. Fascinating. And then from this point is really, can we get into the nanomolar space, right? Like we're a single-digit micromolar, we got pretty good at that, and now we had that information where we knew that extension can get us into that part of the protein that gave us a boost in potency, and we were able to identify, again, through small libraries. The key learning is, I'm not a big fan of large libraries, and they play their role, but I think you can have small focus, 30, 40 compounds that sample chemical space, and there's good SAR built within to teach you what you need to know about what directions to move into. And we got into nanomolar space, and this is where we're starting to see some good biophysical behavior. We're looking here as the affinity of the compound, and it's 160 nanomolar. Starting to see a little bit slower chaos, which is something that we thought would be useful for us. And then this is the SPR version of a functional assay where you're looking at disruption of two proteins, essentially. So we're seeing that they're correlating, which was actually starting to bring us into the nice direction to know that we're moving in the right direction, right? So that you're imparting affinity onto your molecules, and that structural component of affinity function structure, you're starting to now get the desired functional effect out of your molecules. It was 5 o'clock, so we're getting really close to finishing here. So in the end, really, we got down to show that we have some molecules, but as always, if they're nanomolar, they didn't show cellular activity, and then we had some weaker compounds that we can actually show that we can demonstrate cellular activity. It's one cell line that I'm showing biomarkers early. We had some hypothesis of why some compounds, even though they're more potent, we think just have to do with their overall profile. This is, you can spend a lot of time trying to optimize your molecules. And ideally what you want to see multiple cell lines, multiple biomarkers, orthogonal, equate into showing the functional effect that you want to have. So overall, tons of learning from this, and we felt like this was a good springboard to show that we have functional activity, we have some beginnings of cellular activity. The overall profile of the molecules, not great. There's a lot of red. That's really all you need to know on there. I mean, we can talk a lot about, what those numbers mean, but gave us a good understanding of some things that we can do with these molecules, right? So one of the most important thing was where we can pick up our affinity, where we knew we had some room to modulate properties and go back and modify that. We like this turn motif that was solvent exposed and give us some opportunities to think about bifunctional molecules and look at degraders as an opportunity. And that's something that was explored once I left. So yeah, I'll leave it here. This is just summarizing kind of getting from one series to another and what the project delivered in the end during my stay there. And have to thank everyone here. This is all the folks that I worked with, not including my, during my PhD and postdoc, but also the large team that I worked with at the Broad that played a huge role in getting this. And last but not least, my family that really endured through all of these. And yeah, thank you guys for the attention. And again, for the opportunity to come here and talk to you guys about some of the stuff that I've done in my experience.
Thank you very much. That was great. Anybody have any last questions for Cristian before we?
Automation and that it's gone so much to the point that there's such a pile of apps, like apps can literally send reagents or ask for reagents like a centralized test lab full of instruments. They upload their methodology like program, and all those instruments are just run by a small handful of technicians. It's doing all their experiments for them. So going to automation still could that start making the labor more lighter?
Yeah, I mean, there's a big part of our day that's automation. I mean, we have, at least at Novartis, where I was at the Broad, there wasn't a ton of, there was more automation on the screening side, not necessarily on the synthesis side. So yeah, there's tons of that. We do a lot of outsource. We have dedicated library resources at a place like Novartis, so that's all they do. From purifications to library designs and library builds, depending on different methodologies, from all sorts of different chemistry. Photocatalysis is making a huge impact now. So you can do it in micro scale, you can do it in macro scale and whatnot. You can do it in focused libraries, working with enamine or Wuxi. I mean, that's definitely blowing up. It's sometimes is a numbers game. So automation gives you numbers, but I think in cases like this, oftentimes you learn just as much from not having to automate and make 1,000 compounds. You can just learn from a lot fewer compounds. You also have to think for each compound you make. I mean, I heard this number, I don't know how real it is, but it's like for each compound you make in a company, it's probably costed like they equated to some $5,000 to $7,000. So if you don't have to, I mean, this cost savings. If you don't have to make thousands of compounds and you just make 50 to learn the same. So automation plays a role, but I don't think it's going to be the answer of everything. Yeah, it decreases the workload, but at the end, it's still about really making the right molecules to answer the right questions.
Anybody else? One last question?
Yeah, sorry. Went a little late.
Is that Fragment story from the Broad published? Can I read it, see the structure?
I wish. My sources tell me that it's close. There's patents being written and stuff like that. And whether or not there's publications, what happens with a lot of the stuff in pharma as well is people move on, and the people that have worked, I don't think there's maybe one single person that's worked on that project that's still there. So you're losing a lot of the sort of history and information over there, and that probably suffers a lot. I mean, there's a ton of so much impressive science that happens that just never hits the public domain just because it just doesn't get published, so hopefully, but I don't, patent, I think you'll see for sure whether or not there's going to be a follow-up publication with more detailed information. I don't know. So, if it does, I'll send it over.
Well, without further ado, thank you very much. That was fantastic.
Yeah,
Great to
thank
have
you. It was fun to do this. Maybe go cancel it.
Beautiful. Thank you.
Yeah.
And Miller.
And bad.
Yes.